Let's look at how plants changed over time so that they were able to move from the marine or aquatic environment onto the more challenging terrestrial environment. First, let's review the process that occurred within the organisms that lived in marine or aquatic environments. The first organisms that were photosynthetic were blue-green algae that were prokaryotic in format. As organisms increased in size, a blue-green algae was entrapped in one of these larger organisms, and the blue-green algae served as the precursor of the chloroplast. The glaucophytes present muroplasts that more resemble blue-green algae than the chloroplasts of higher plants. The next progression had rhodoplasts, which have phycobilisomes that are red in color and therefore impart a red coloration to the organism. Progression then moved into the true green chloroplasts, which are found in the green algae. From that point, the organisms began to get more organized and began the development into the higher green plants. There are several advantages for photosynthetic organisms that were able to make the transition to land-based environments. First, there would have been more light and light energy available to photosynthesis, as air is more transparent to light than water is. Carbon dioxide concentration in water is approximately 386 parts per million, but diffusion in water is about 10,000 times less than that of air. Oxygen concentration in water is about 8 parts per million. On the other hand, carbon dioxide concentration in air is about that in water, but diffusion is much higher. Oxygen concentration in air is about 21%, or 210,000 parts per million. Also, terrestrial soil is much richer in nutrients than is aquatic soil. This is because the water above the aquatic soil dissolves nutrients and therefore removes them from the soil. With respect to modifications in photosynthesis, muroplasts have peptidoglycan in the wall. Peptidoglycan is the polymer that is found in the cell wall of bacteria. Phycobilisomes are also present. These are light-harvesting antennae complex of photosystem II and are red in color. The wavelength that are absorbed is in the 500 to 600 nanometer range. Rhodoplasts found in red algae allow photosynthesis down to a depth of 268 meters. Rhodoplasts also store energy in the form of Floridian starch, which is an intermediate between amylopectin and glycogen. They also have pyrenoids, which help in carbon dioxide packaging. The final step up is the chloroplast, which is green in color and do classical photosynthesis. There are 4 major developments that have occurred to permit the transition from marine, aquatic environments to land environments. The first is embryo retention. Algae release gametes into the water, and therefore the zygote is formed in the water. Higher plants retain the embryo in the plant. This provides for additional protection during the development of the embryo during its initial formation. This is a picture of a dissection of a fruit showing the seeds which contain the embryos. All are internal. This is the only adaptation that is shared by all plants. Another adaptation is the cuticle. The cuticle is an impermeable barrier on the surface of the leaf that prevents desiccation. The cuticle is composed of polymerized fatty acids and therefore is impervious to water. The initial concept of cuticle will provide protection from sea salt spray and later became more adapted on plants to protect from desiccation. The cuticle is thin on bryophytes. This photo shows the cuticle as an outer layer on the surface of the leaf. Another adaptation is the formation of stomata. Stomata permit gas exchange. This allows for regulation of processes. Bryophytes have minima stomata. They are fixed, and these are mostly found on the sporangium. These pictures show the leaf of a moss without stomata, the stomata on the leaf of a fern, the stomata on the cross-section of a pine needle, and the stomata on a flowering plant. Note that the stomata are variable based on guard cells. The vascular system is also imperative to creating larger plants as it permits the transport of water from the soil to the upper reaches of the plant. Algae do not need vascular tissue as they absorb directly from the water. Bryophytes are also loosely developed and have no lignin. The leaves are thin and only a few cell layers thick. They do present a hydroid, which is a xylem-like tissue, as well as a leptoid, which is a phloem-like. These are not yet considered true vascular tissues, but they are important in translocating material even in these small plants. Above these, the plants do present a true vascular system. Here is a photo that illustrates the location of phloem and xylem in a higher plant. The progression from marine aquatic environments to the terrestrial environment require a few distinctive changes that occurred and that can be traced in higher plants.